your body that you can string together to make proteins. They're strung together in a very specific sequence. Sometimes a protein will tolerate one being off, but it's very, very specific. They range from 100 to 10,000 amino acids long. And the average protein is 150 amino acids long. Okay, so if we're going to string together 150 amino acids, first of all, in all your DNA, all the C's and the G's and the A's and the T's, there are 3.2 billion of those in your body, in every cell. 3.2 billion. So if we have an average protein of 150 amino acids, if insulin has 84 amino acids, it's one of the smaller proteins in your body. Okay, so insulin, if you have 84 that you have to string together, and there are 20 amino acids that you have to choose from to put in there to string that together, the chances of that happening are 20 to the 84th power. Or, something we're more familiar with, that is equal to 10 to the 109th power. That you could take that box of 20 amino acids and put it up and put 84 of them together, the chances of getting any particular poker hand there is 10 to the 109th. That's a small protein. Okay? Does that make sense? Okay. Next slide, Danny. Okay, so proteins are coded in genes. And you might already know this, but let's suppose, this thing is going to be a model for a lot of different things tonight. But right now, we're saying this is one strand of the DNA. One strand, and we're going to say that it's 3.2 billion long. Okay, it goes all the way down to Mickey's really small, tiny garden out there. And where's Mickey?
I'm waiting for her to talk about CNA.
Okay, so it's, see, now you made me lose my place. Okay, so if we have this long strand, from here to here would be a gene, and that's what codes proteins, from here to here. Okay? In between the genes are vast amounts of junk. Nothing's there. Okay? And the gene, from here to here, is also divided up into exons and introns, and I'm going to explain that in a little bit here. Okay, so genes are a section of a DNA that is the coding instruction for proteins. It's the instruction manual for the sequence or the order to assemble amino acids into proteins. You have about 20,000 genes in humans. However, DNA and proteins speak a different language. RNA is the translator. Okay, next slide. So we've got DNA makes RNA, and there's two types of RNA, a messenger and a transfer RNA, and then that makes proteins. Okay, so this is where it goes from DNA to messenger RNA, right here. If this is now a double strand, then this molecule called transcriptase is going to come and clamp down on this. And it's inside, that's this red box here. Inside this enzyme, by the way, which is a protein that had to be made by this system. How did that happen? Okay, so you've got the transcriptase here. It takes and unwinds the DNA. And it unwinds it, and then new base pairs come down and go into this molecule and start to make a chain up here of a copy of this one, of the bottom one. Because here's a G, there's a C down here, a T and an A, so that has to match up. So it's going to make a copy of this one. That's how it makes messenger RNA. Then it zips it back up. Okay, so how does your body know if it has to make insulin, for example? How does it know where to go on that 3.2 billion? How does it know where to go in order to do that? How does that happen? So you were hoping I was going to tell you? Okay, well, we're going to talk about that. Okay, so that's the messenger RNA. We made a template. We made a copy of the DNA. That was done in the nucleus. We don't want to do this out in the middle of the cell because then we're risking our DNA. Something might happen to it. We want to do it in the safe confines of the nucleus. So then this messenger RNA goes out into the cytoplasm, and it gets changed. Okay? It goes into the ribosome. And here's the messenger RNA. We've got another molecule that clamps down on the messenger RNA. That's the red box here. And what this does is it takes, this is a molecule. There's 3 on one side and 1 on the other. Okay? The one on top is an amino acid. That's how we get to the amino acids, because these are just base pairs. Okay, that's the DNA. So, if we have a CGA, this is going to come up and it's going to match that. And then, as we go along, these amino acids are going to line up in a chain. They're going to line up. So we've got our messenger RNA, and then our, this is called a ribosome. And that is going to come, and it's going to attach on there, and then the amino acids on top are going to form a chain, a protein chain. These 3 molecules are called codons. Okay, and that's the reason, is because of this molecule. It has 3 on there. Okay, so it's the code. When they talk about the genetic code, that's what they're talking about. It's the code that translates this into amino acids. Okay, next slide. Okay, I totally lied to you. Okay, this is where you need to start paying attention again. Okay, because Mickey,
Yes?
if I don't tell the whole truth, is that a lie? Come on.
I'm not going to say anything.
No?
Until after you're done. And yes, it is a lie.
If I don't, okay, then I lied. Okay, because there's much more to the story. Okay, that shows up there one of these, messenger RNA. How does your body know where to go in order to start that? Okay, so you can see, if my pointer will work, we've got translation start and translation stop. That's a very specific codon right here. It's very specific base pair. So when that molecule is going over and attaching onto here, it has to attach onto the translation start. That's how it knows where to start. So not only do we have to get our 84 amino acids in a row, okay, and if there's 84, let's use our 150, because this is the average size of the protein. If we're going to make a protein with 150 amino acids long, we have to get 450 times 3, because of the codon, base pairs in a row. 450 base pairs in a row to make that protein. That's pretty amazing, isn't it? That have to be in order, in sequence, to make one protein. Okay, so it's not 150, it's times 3. That'd be 450 that you have to get in a row. Does that make sense? Yes? Okay, so now we know that you have to get those 450 base pairs in a row, but you also have to win the Powerball. Not only do you have to win the lottery, you have to win the Powerball, not once, but twice, once on each end. Right? I mean, that's basically what you've got to get that one right, or it's not going to start. You have to get that one, or it's not going to quit. Okay, so the other thing is, how does your body know how much of this to make? How does it know when to stop making it? And molecules are not start and stop. They're regulated, and their levels gradually change. Sometimes your body makes the chemical, and it will store it for a while. How does it know when it needs to store it, or whether it... Okay, that's all done by regulation proteins, by proteins. That's what this is. This is before this start, we've got enhancers and promoters and what's called a TATA box that regulate this procedure on how to do this. Okay, then I lied additionally because we've got introns and exons in here. Exons are the ones that code for protein, not introns. And the average gene has 8 exons and about 9 introns, the average one. So it's not that it just goes down the line. It's interrupted by the introns. And I also said that between the genes was junk and that the introns was junk. Okay, it's not junk. What your body does, next slide please. It takes, and it has a molecule that might be the most amazing one of all. It's called the spliceosome. And it attaches onto this, and it splices the introns out. It takes them out. Okay, so this molecule, this intron, will go up into a bow when that molecule, that spliceosome, attaches onto it. And it takes it and it snips on each end, and then it takes the two exons and attaches them together. Okay, so it's called splicing, and then it's protein synthesis. The spliceosome, every time it takes out an intron, it disassembles. And then it reassembles for the next one. Then it disassembles and it reassembles for each protein that you make in your body. Okay, so when I go like that, there's noise. Your body's making proteins right now. You're hearing that, it's going up into your brain, chemicals are changing, your body is instantaneously making proteins to interpret that. Then, when you go to sleep tonight, it's going to rearrange it and it's going to put it into long-term memory. All that's done with proteins. That's all so that, next year you'll say, remember when that crazy guy slapped his hand on the table? And you pull that up, that's all done by proteins and that's all how this is done. When it's pulling it up, it's making proteins to snip and to splice all this. Okay, next slide. This is the DNA code. When you hear about the DNA code, this is what it is. This is the chart for it. We've got our codon, and this is first letter, second letter, and third letter. Let's suppose the first letter is C. Then we go and define C, and that's the first one. Second letter, let's say it's C. Let's say the first letter C and then U and then A, just to make it easy. Then leucine, that's the amino acid that's on top here. Okay? Does that look like it was designed? It sure looks like it to me. I don't know. We've got 3 stop codons up here. We've got 1, this is a start codon. Okay, so when AUG is there, it starts. Now you would think if it was done randomly, that you'd have just as many stops as starts, I would think. But it's not. There's 3 stops and 1 start. So if you just go and you find the first letter, the second letter, and third letter, and you can tell what amino acid's going to be put up there. Next slide. Okay, we already went through this. This is the codon, and right there, attaching on the 3 base pairs, and there's your amino acids that are forming. Next slide. This is the transcriptase. Okay, and the reason I put this up there was because they just figured this out like a month ago. This just came out from a study about a month ago. They got a 3-dimensional picture of what that looks like molecularly, of the one that transfers it into messenger RNA. These are proteins. These different colored things, those are proteins. That's a protein, this is a protein, that's a protein. All these are proteins. And that's what that molecule looks like that does that amazing thing. Okay, so I know what you're thinking. You're thinking, wait a minute. He told me that this is a long string, that these proteins are long strings. That is not a long string. Okay? Is that what you were thinking? No? Okay, so we're not done making the protein yet. Okay, next slide. Proteins have to be folded. Okay? They have to be, they look like mangled coat hangers. And proteins, after they're assembled, they have to be folded. Your body only uses left-handed amino acids. It won't use right ones. In nature, a lot of chemicals are right and left-handed. And they call them chiral molecules. And they're different. My hand will not, it's a different molecule. And in nature, they're about 50-50. But your body will not use right-handed. All the food that you eat, all the plants that you eat have left-handed because that's what nature uses. Now this is a big mystery on how that got started, because when life got started, there wasn't a procedure to take that out, to take the right ones out. So, okay, so cells use only left-handed amino acids. There are two proofreading areas in the ribosome here, where if there is a right-handed amino acid in there, they're called scrubber molecules, and they go in and they take it out and they insert the right one. A lot of processes, chemical processes in your body have scrubber molecules, where if the whatever your body's trying to make doesn't turn out right, the scrubber molecule goes in and destroys it, sends it to the lysosome to be recycled. And does that sound like design? It just sounds like. Next slide. Okay, that's what a hemoglobin molecule looks like. That's after it's folded. And these are families of proteins here. The blue would be a family of protein, a certain type. This is a certain type. These are certain types. And they're all strung together, and they're folded in the ribosome here, in a complex that is not even remotely understood. Next slide. This is actin and myosin, which are your muscle proteins. We've got this part and this part. Those are two different proteins, actin and myosin. Next slide. And this is what the ribosome looks like. Okay, this actually separates right here, and then that's how it clamps onto the messenger RNA. Next slide. So the ribosome folds the protein in a very specific manner. It's like a lock and a key. Only certain molecules can fit into that ribosome. If they don't fit, they don't get in. That's how your body controls those chemical reactions. It labels the protein, the ribosome. When it makes the protein, it folds it, it labels it with a destination label. It puts a marker on it to tell your cell where that is supposed to go. It can attach on a specific sugar or a specific metal to the protein. There's an amazing quality control mechanism that sends incorrect proteins to the recycle bin. And most, when they are finished, are sent to the Golgi apparatus for storage in a vesicle. Next slide. Okay, the Golgi apparatus packages the proteins into packages called vesicles. Then it delivers it, a lot of times, to the, if this is your cell, to the inside border of the cell wall, the plasma membrane. Or it takes it outside the cell, or to the lysosome to be recycled. They compare it a lot of times to a supermarket bakery. Next slide. Because it receives bulk flow. It goes to an unloading bay. The goods are received. When they're received, those proteins are checked over. Some of them are sent back. The good ones are stored in an orderly sequence, okay, according to the label that they have. Some of them are converted to another product and then relabeled. Some are transported. If it has to get around in the cell, it's transported on a microtubule by a molecule called kinesin. And Danny, can you start that? We've got the movie. This is only like 22 seconds long, okay? It's on YouTube if you can get this going. This is what the kinesin molecule looks like, and that big blue thing is the vesicle. That's how it transports it on that tube. So look here, that's what that molecule looks like. It's transporting, that's what it looks like. That's transporting things in your cell right now as you speak. Those strings going through there, thank you, Danny. Those are microtubules. They are self-assembling. Because if they were there all the time, everything would get in the way of each other. So if you have to move this over to here, that will pull on that kinesin molecule, and then as it goes along, the microtubule will be built in front of it until it gets to here, and then it starts taking it down on the other end. It's just amazing. I don't know, it's just amazing to me. I just can't. That's an animation, of course, but that's actually how